Hi everyone, my name is Mikael Malmqvist and I'm a principal scientist in the Research and Development Department here at Atlas Antibodies. Today I will be giving you a tutorial in how to perform the image processing and image analysis steps that are needed to analyze the MolBoolean results. I will be sharing my screen throughout this tutorial, showing you all the steps, and I will be turning off my camera now so that I can make the full use of the screen. Content in this tutorial is also described in the MolBoolean instructions for use, and these can be found on the MolBoolean product landing page here. On the MolBoolean product landing page, you will find a short description of the product and some example results in cells and tissue. But also at the bottom of the page, you will also find user instructions and tools needed for image analysis. So you can click here, user instructions, read user instructions to get to the bottom of the page. And here you can download the MolBoolean instructions for use and product data and safety sheet. You will also find tools for image processing and image analysis. We will get to this later. Tutorial, we will be using two examples, and images from those two examples are located in the appendix of the MolBoolean instructions for use. The first example is where we have performed MolBoolean staining of E-cadherin and beta-catenin in MCF7 cells. These two proteins are known to interact in the plasma membrane of cells. However, not all of these proteins are interacting with each other. Some are also present as free non-interacting proteins. So we will quantify the percentage of free versus interacting protein fractions, and I will show you how to do this today. The second example is where we have performed MolBoolean staining for the same two proteins, E-cadherin and beta-catenin, but in human tissue. MolBoolean analysis of tissue images may differ slightly from what is done in cells, and I will be showing you how to handle tissue images today as well. To help achieve an efficient segmentation of rolling circle products in the MolBoolean image analysis pipeline, we first need to perform a post-image processing called deconvolution. When an object is imaged with a microscope, it will appear slightly blurred with a certain noise around the edges of the object. This is called convolution of point objects. And the amount of blurriness is determined by a point spread function that's called PSF. Image deconvolution removes the noise around the objects in a microscope image and thereby improves their resolution. Image deconvolution can be performed using several image analysis programs, for example, Huygens deconvolution by scientific volume imaging. Often these softwares or programs require a license cost. Otherwise, many of the microscope image acquisition softwares nowadays also include integrated deconvolution options. But image deconvolution can also be performed with the freeware image J using a certain deconvolution plugins. And I will be showing you how to use image J today to perform the deconvolution. Instructions on how to download image J and the two necessary plugins needed to perform deconvolution can be found in the MolBoolean instructions for use document that is provided on the MolBoolean product landing page here. The image J software first needs to be downloaded from the imagej.net website. You do download it either Windows or if we're on Apple computers. When you have downloaded ImageJ, and you need to download two deconvolution plugins. and you need to install them in the ImageJ interface. You can get the plugins from the MolBoolean product landing page. And when you've downloaded ImageJ and 2 plugins, you have to open the ImageJ application here through the shortcut in the taskbar. So an ImageJ interface will appear. You should see 2 new entries in the ImageJ plugins menu: diffraction PSF 3D and parallel spectral deconvolution. And we're going to use the 2D spectral deconvolution. So these 2 will have to be installed according to the MolBoolean instructions for use before we can use them. So first, you will have to create a theoretical PSF image. As mentioned earlier, when an object is imaged with a microscope, it will appear slightly blurred with a certain noise around the edges of objects. The amount of blurriness is determined by a point spread function, PSF. If we know the PSF for the microscope settings, then we can perform deconvolution on the images. So first, we need to create a theoretical PSF based on the microscope image acquisition settings that we used when imaging the MolBoolean slides. So now we're going to create a theoretical PSF image for each fluorescent channel that has been used during image acquisition. That means that for MolBoolean analysis, 3 different PSF images will have to be generated: the PSF Atto 565, the PSF Atto 647N, and PSF Hoist 33342. That's the nuclear counter stain that we used in our example, but you can also use DAPI in the nuclei. It's important to remember that these PSF images that we're now going to create will only have to be generated once for the same image acquisition settings. So if you always take your images with the same settings, then you can use the same PSF images every time. So first, you will open the diffraction PSF 3D plugin. And in this window, you will find 11 input fields that need input to be able to create a PSF. And I'll walk you through them now. So the first is the index of refraction for the media, the immersion media. So that's the immersion medium that was used on the microscope objective during imaging. If no immersion medium has been used, so if you use a dry objective, then the immersion medium will be air and the refractive index will be 1.0. But often you use some sort of immersion oil on high magnification objectives. And so you'll have to find the refractive index for that immersion medium. Often that is printed on a bottle or available online. So, for example, if you use a conventional microscope immersion oils, the refractive index will be 1.51. But in the example images presented in this protocol, we used water immersion. So therefore, the refractive index in our example for water is 1.33. So we enter that in the field. And then we have the numerical aperture. And that should be printed on the objective itself, along with the given magnification. In our example, we used a 63 times objective with a numerical aperture of 1.2. The next thing is the wavelength. So, and that should be entered in nanometer. And that's the maximum emission wavelength for the fluorophore. So in the first case here, for Atto 565, the maximum emission is 590 nanometers. So you put 590 in there. Then you have the longitudinal spherical aberration. So you simply press 0.00 there. And the next is the image pixel spacing. And if a confocal microscope was used, this is the pixel size for the generated image and should be entered in nanometers. The pixel size can be found in the image properties. So in our example, we acquired the image with a pixel size of 51 nanometers. And so we used a Leica SP8 confocal laser scanning microscope to image the MolBoolean stain slides. So here we have the images in the LAS-X software that is connected to our microscope. So if you right-click on one of the images, that in this case, you get takes sometimes. Here we have it. It took some time to load it in this case. Right-click and then you go to Properties. And here you can have, I mean, there will be different for different, obviously, microscope softwares, but for Leica, you have you can get the pixel size here that was taken. It was used during image acquisition, so it's 0.051 micrometer, and you have to convert it to nanometer. So in our case, it was 51 nanometers. If a wide field microscope was used, and image is required with a camera, then you can find more instructions on how to calculate the pixel size in the MolBoolean instructions for use document. The next input is slice spacing. And this number is important during 3D deconvolution, but it's irrelevant for 2D deconvolution. So we put 0.00 here. And so it's important to note that this protocol what I'm describing today will only outline the process for 2D deconvolution. Even if 3D Z stacks have been acquired, I recommend that you deconvolve each slice of the Z stack individually, and I'll talk more about this later, why it is important. Then you have the width and the height in pixels of the acquired image. So in our case, we used 2400 by 2400 pixels. So again, you can find that in your properties viewer image in your microscope software. So here, logical size 2000 in X and 2400. So 2400 in X and 2400 in Y. Then you have the depth in slices, and to the deconvolution, we only have one here. Keep the next input normalization to the default sum of pixels values equals 1. And then the title of the PSF, it's just how you want to name it. So I recommend you name it PSF for the 565 channel. And then basically just press OK. And OK again. And it'll take a few seconds. And you will then generate your PSF 565 channel. So if I enlarge this now, you'll see that there's a black image with a small white dot in the middle, and that's your point spread function for the 565 channel. So we're going to save this as a TIFF somewhere on your computer. So save as. I recommend that you create a folder on your computer where you're having certain subfolders. One's called PSF. So if you open this PSF folder, you can then save it as a TIFF, and you save it at PSF 565. I've already saved these already, so I will not save this particular one. Now repeat the same procedure for the Atto 7N and the Hoist 33342 or DAPI, if you use that instead, fluorescent channels. So the only detail that needs changing in the input fields is the wavelength and the title. So if you were to change to a PSF647N, you need to change this to 664, which is the maximum emission wavelength for ATO 647N. And then you can change the title name. And you get OK again. And you get your PSF647N. And you save that as before. The last one is, like I mentioned, is the hoist. And the wavelength for that is, the maximum wavelength is 461. If you used DARP instead, the maximum wavelength is 465. In our case, it's 461. We just more than white dot there, obviously. Save that as before, and then you will end up having in your folder for PSFs, you'll end up having 3 different, one for 565, one for 647N, and one for hoist. The second step is to perform the deconvolution of the raw TIFFs from each fluorescent channel against their respective theoretical PSF that we just generated. So this section outlines the process for performing 2D deconvolution. That is deconvolution on a single image focal plane, so a single Z slice. If you have required 3D Z stacks, it is important to perform individual 2D deconvolution on each Z slice so that they can be analyzed separately in Cell Profiler later on. The reason for this is that if a maximum projection image of the 3D Z stack is used, there's a risk of creating false positive overlapping fluorescent channels, fluorescent signals, which is then will be classified as protein interaction. So basically having rolling circle products being present in separate Z slices. And therefore, it is important to not use a maximum projection image of the 3D stack. So when you want to perform this 2D deconvolution, the first thing you have to do is to export the fluorescent images from the microscope software. They need to be exported as raw grayscale TIFF files. That means you'll export 3 separate TIFFs per image set. So you'll have the hoist 33342, and that will be as channel 00. The ATO 565 will be as channel 01, and ATO 647N as channel 02. So you will right click on the image, in this case in OSX, and you will export the image. And then you will save it. You export as grayscale raw images, TIFFs. So basically, I will show you how that will look like. But you will do that for each image set in your example. So once you've exported the images, you might have it in a folder called raw grayscale TIFFs. In the same folder as where you have the PSF. In here, in our example, we will have our list them. So basically, you can see that there are 3 sets of images, 3 channels per set. And if I list them with details instead, we can see that we have 3 channel 00. That's the nucleic counter stain. And there will be 3 with channel 01, and that's the ATO 565. In our example, that's the E-cadherin antibody, the mouse E-cadherin antibody. And then you have 3 channel 02, that's the ATO 647 channel, staining coming from the rabbit beta-catenin antibody. that we used in our example. So when we have exported our images, we have created our PSF images. So now we want to perform the 2D deconvolution. So the first thing is to open your PSF image. So your file, open. And in the folder where you saved your PSF images, here, you will then click on, for example, in this case, we have to do every channel separately. So we start off with the 565 channel PSF. And then we need to open the grayscale TIFFs that corresponds to the Atlas 565 channel. So that's the files ending with the channel 01. Open. Go back to your raw grayscale TIFFs. Open. And here I will open all the images that are ending with channel 01. Open. So that's your raw grayscale TIFFs for the Atlas 565 channel. You'll have image 3, image 2, image 1. Oh yes, I forgot to mention that it would be good if you, when you save the images, you can obviously use whatever file name you want. But in the example shown today, the different image sets are called image 1, image 2, and image 3. So you'll see that there are 3 different channels per image set. So in this case. So once you've opened your PSF and your 3 grayscale images for that channel, you go to plugins, you go to parallel spectral deconvolution and 2D spectral deconvolution. Click and a new window will open with multiple drop-down menus available. And in the image drop-down menu here, you will select the image that you want to deconvolve. Start with image 1, and in the PSF drop-down menu, you'll select your PSF 565. And then you will keep all the other settings. Keep the default settings. Don't change anything here. And you just press deconvolve. So that's your deconvolved image appearing after a little while. So I would put the original image next to the deconvolved, so you'll see the difference. You can see that the raw image is definitely the rolling circle products have become much more defined into clear, sharp objects, and you also increase the intensity slightly in the image. So you will want to save this deconvolved image. I'll save as TIFF. Everything is saved as TIFFs in our case. I've created a folder called deconvolved images. And basically I'll save it exactly with the same naming as the raw images, but just add decon between image and channel. So I know that it's the deconvolved one. And I've already saved them, someone saved this one. And then you will do exactly the same thing, but all you do is change the image to image 2, and you do deconvolve again, and save that one. And then you do image 3, and deconvolve again against the same PSF. So then you end up with 3 deconvolved channel 01 images in the end, as you can see, 1, 2, 3 for the 3 different image sets. Now you'll have to repeat the same procedure for the Atto 647N. channel, which is channel 02, and also for the hoist 33342 images, which are channel 00. So you do exactly the same thing. You open up the PSF for the 647N instead, and you will then open up your 3 channel 02 images. So I won't go through this also in the interest of time, but it's exactly the same procedure. So in the end, you end up with 3 deconvolved images per channel, 3 different image sets. Once image processing is finished, we need to perform image analysis. And image analysis is needed to calculate the fractions of free versus interacting proteins in the MolBoolean results. An important step in the image analysis workflow is to achieve a good segmentation of the rolling circle products. And we call the rolling circle products RCPs. So this segmentation allows for the specific quantification of the number of RCPs that are detected in only the ATO565 or only the ATO647 fluorescent channels. And these represent the free non-interacting fractions for the 2 analyzed protein targets. But it also enables to quantify the RCPs that are detected in both the ATO565 and the ATO647 fluorescent channels. So if you have RCPs that have fluorescence in both channels, that will indicate protein interaction or proximity in the sample between the 2 proteins. Any image analysis software that can achieve such segmentation and then quantification can be used to analyze the MolBoolean results. One image analysis platform that can be used to analyze the MolBoolean results is the freely available cell profiler image analysis software, and developed by the Broad Institute of MIT and Harvard. The first time MolBoolean image analysis is performed, you will need to download the cell profiler software. And instructions for how to download cell profiler software can be found in the MolBoolean instructions for use. And we have created a cell profiler pipeline dedicated to the MolBoolean analysis. And the pipeline is available for download on the MolBoolean product landing page. So you just basically click here, and you'll download the MolBoolean cell profiler image analysis pipeline, and you'll save it that file somewhere on your computer. So here we have the MolBoolean image analysis pipeline saved. So first, you'll have to download the cell profiler software from the cell profiler website. And then you'll have to download the cell profiler pipeline from our product landing page. And then if you want to open the pipeline, you just double click on it. Pipeline takes maybe a minute to upload, and what you will encounter is this black window where you set telling you the starting cell profiler. But also you'll have an error code here. And don't worry about that error code. Basically, it's just is due to the fact that we've added some instructions in the pipeline in text format up here. So don't worry about that. So you close that window, this black window. You would also, when you open the pipeline and get this welcome to cell profiler window, you can basically just close that window as well. So here you have the cell profiler pipeline, and I will walk you through it now. Basically, cell profiler is built up of modules that are going to be used to analyze your images. So you can make up your own pipeline by just adding modules that you want to do. So we've made a specific one that is dedicated to the MolBoolean image analysis. So what you have to do is you have to upload your images by dropping your files here. And so we have our deconvolved images that we want to use. So you locate them in your in a dedicated folder for that. So here we have all those deconvolved images. I just highlight all of them and drag them into here. So you end up with 3 image sets, 3 channels per image set. And each image set is called image 1, image 2, image 3, as you can see here. And each image set would have this channel 0, 0, channel 1, and channel 0, 2. Then next, if you click on the metadata module here, this gives you the option of extracting information from the image file names, so you can extract metadata, and that will be stored along with your measurements. The metadata to be extracted is defined using this regular expression input field. As a default, the MolBoolean image analysis pipeline extracts the image number from the file names. So you can click on this magnification glass here. You can actually put in your own regular expression here, but in default we have the image number that is extracted. So you change this as you please, depending on how you name your files. But once you just need to, if you're using the default settings, or even if you change it, you need to click update down here, and that's done. And then you move on to the names and types module, and here you're gonna assign names to the image files that are originating from the different fluorescent channels. So to all the channel 0, 0, we can assign names. As a default, we assign that to DNA. All the channels that are called channel 01 have the name Atto565, as I mentioned before. And the images that are from channel 02, ending with channel 02, we're given the name Atto647. So as you can see that any file that contains this underscore channel 02 will be given the name Atto647. So when you press update down here, the channels will be assigned to these names. So channel 01 will be Atto565, channel 02 will be Atto647, and channel 00 will be DNA. You see I've actually dropped the N on the end. So the fluorophore that we use for MolBoolean is Atto647N. But just to make it easier to label, I've just labeled it Atto647 in this case. The MolBoolean image analysis pipeline is composed of a series of modules that will ultimately segment and quantify the number of RCPs with fluorescent signal in the Atto565 or the Atto647 channels. The modules are preloaded with the default settings. However, these settings may have to be altered slightly based on the image parameters of your experiment. Cell Profiler permits the testing of your settings in test mode before performing the final image analysis. So in the test mode, you can adjust some of the settings so that they suit your images before you actually do the final image analysis. And I will walk you through these settings now. So basically just click start test mode down here. And so the test mode has now started, and the current module that is being used in test mode is indicated by a green arrowhead here. And to visualize the output from a module in test mode, you have to make sure that this eye is highlighted, and that you have a green tick on that module. I will now walk you through the modules that should be tested in test mode at least the first time you're analyzing your images. So first you have the identify primary objects. So the aim of this module is to select the number of nuclei in the images So at first, keep the default settings and click step down here. You're not going to do that yet. But this will then open a new output window showing the identified nuclei in the image. So we press step now. I want to mention that this could take slightly longer now that we are recording, so bear with me. I might even pause the recording to fast forward this for you. The cell profiler opens a window showing the identified nuclei in the image, so I'll enlarge this output window. So this window will display the input grayscale image here in the top left, and that's your channel 00 for the first image set. And they will show you how it says selected nuclei based on your settings. And basically will show you this nuclei encircled in green here, and how they've selected that particular nuclei as well, and counted the number of accepted objects. You will also notice that when you have clicked step down here in Cell Profiler test mode, you move on to the next module automatically. So the green arrowhead here will move to the next module. If you see that some of the nuclei were not selected in the output window from the first module, then we can make some adjustments to the settings in that first module. So you could adjust the typical diameter of the objects. Here, the default is with minimum 200 and maximum 550. And you want to make sure that each nuclei is only counted once. You don't want to have substructures counted within the same nuclei. And you could actually use this measure tool. And if I enlarge the window again and you click this measure length, you can measure the diameter by hovering over, clicking on one side and then dragging to the other side of the nuclei. And you'll see the length down in the right-hand corner, length. You can also get some sort of idea of the sizes of the nuclei by looking at the median diameter down here. Basically, you don't want to use a minimum diameter any less than the smallest nuclei in your image, because if you do any less, then you're running the risk of picking up substructures within one nuclei. Another setting that might need adjusting, if you're still not picking up the correct number of nuclei in your image, you can change this 2-class or 3-class thresholding. So the default setting is to use 2 classes, but you can change it to 3 classes, and you can try either foreground or background here. When you have changed any settings in Cell Profiler, you can get an updated window by clicking step again here, and then you'll get the same output window, but with the nuclei selected based on your settings. But in our case, the default settings manage to generate the correct number of nuclei. You will also find that if you are updating some settings in the module in test mode, the profiler will go back to that module that you're changing your settings in. So then you have to click through that module again in test mode to get to the next module. So then you click step, or you can also click the green arrow head here. So then I have to go through to click step again in test mode to get to the next module. Another thing I want to mention is that when you are in test mode, and you don't want to use the first image set, you want to look at the second or third or fourth, you can press next image set down here. Then you will walk through the test mode modules with another image set that you've uploaded in your images. Finally, I always want to mention that it might not be possible to efficiently determine the number of nuclei in the images. For example, it can be difficult in tissue images where the nuclei are often less uniform or have varying intensities within the same nuclei. And I will show you how to analyze those images in this tutorial at the end. But basically, then you will not really use this module where you select the nuclei. I'll get to that later. So now we've gone to the second module, which is identify secondary objects, and you see that the green arrow head has moved to that module. But you cannot really, if you want to look at the settings, you have to click on there to see that module here. Again, I recommend using the default settings, but this module it aims to outline the cell borders. And the cell borders will be estimated based on the nuclei as a starting point. So it is within these cell borders that we're generating now that the number of RCPs will be counted later in the respective channels. So when you click step in this module, the profiler will generate an output window showing the outlined cell borders based on the settings. So I will increase this window, size of the window, so you can see that cell profiler has actually moved away from the nuclei and segmented the cells in cell borders here. And it's not counting any cells that are lining the borders of the image. If some cells are missing, you can change settings here to so that here the default setting is using propagation to move out from the nuclei. You can actually select distance n as well, and use a number of pixels that you want to extend from the nuclei. If you wanted to use this instead, you again, because you've changed the settings now, have to press step again to generate a new output window. But in our case, the propagation works well to segment the cells. Again, following on from the comment I made earlier for the first module, if you can't select nuclei properly in the first module, then you won't be able to segment the cells properly either. So again, if you're using, for example, tissue images where it's difficult to determine the number of nuclei, it will be very difficult to determine the cell borders as well. And I'll show you later how you can do to analyze tissue images. Now Cell Profiler has moved on to the next module, indicated by the green arrow head, moving on to the Enhance or Suppress Features module. So you can click on that to see the settings for that module. And this module is performed for both the Atlas 565 and 647 channels. So you have two copies of that module. It aims to sharpen the RCPs in the images. And the module is not necessary if images have been deconvolved. However, for the functionality of downstream modules in the pipeline, it is still important to just run the images through these modules. And the result window, when you click step, will display the raw Atlas 565 channel image to the left, and then the enhanced image that is named filtered Atlas 565 to the right, in the right field. And you will aim to select a feature size here that generates a filtered image that is unaltered compared to the original input image. So you don't want to change the images now because we already have a good sharpness in our objects from the deconvolution. Later on in the test mode, we'll get to a module called display histogram. So basically you measure the object sizes of all of the RCPs, and then you display a histogram of the different sizes that you have for the object in your image. And that will give you an overview of your sizes of objects in your image. Once we've looked at that, we can see the largest RCP in our image, and then I think it's recommended to add the largest the size of the largest object as a feature size here. So we can go back to this module later on and alter this if we have a different feature of the objects. And so basically then you will do the same thing for the 647 channel. So you click step on that one too. Once you've gone through the enhance and suppress features for both the Atlas 565 and 647 channel, you move on to another module called identify primary objects. I'll just click on that to see the settings for that module. This module is performed for also for both the Atlas 565 and 647 channels. So you have two of those. It aims to identify and segment all the RCPs in the image. At this stage, you keep the default settings and move on to the next module by clicking step. Adjustments will be made later when we know the maximum and minimum sizes of the RCPs that will be displayed in this display histogram. So for now, we just click step for both and keep the default settings. That's the first one. I will walk you through these results very soon. I'll click the second one also, identify primary objects for the 565 channel. Click step on that. The results for the 565 channel. Again, I will mention this later. Just want to mention that all your result windows are actually available down here. If you haven't unclicked them, obviously close them down. But if you're just minimizing them, they'll be down here. If you hover over the cell profiler logo down here. So now we're in the measure object size and shape. Here we're just basically measuring the sizes of the objects in the 647 channel in this case. The RCP objects will be of similar sizes in both channels, so it's not necessary to measure the sizes for both channels. We can just use one of the channels as a measurement of sort of get an idea for the shapes of the rolling circle products. And so here I'll just press step. Note that there won't be any output window from the measure object size and shape because we don't have an eye highlighted here. And so we will just move down to, when it's finished, we'll move down to display histogram. And here we will then display the sizes that were measured in the measured object size and shape. So you just click step on that one. And you will get an output window showing you the distribution of the sizes of the rolling circle product objects, RCP objects in the 647 channel. So you will see then the smallest sizes and the largest sizes. And to get a closer look of the scale on the x-axis, you could, in any output window really, you could use this magnification glass, and you can highlight this area here. And then you can increase the magnification and you will see the x-axis here. So you can then decide on the smallest object in your image. So in this example presented here, we can empirically decide that 3 is the we can average up to 3.5 or below to 3. It doesn't really matter. But let's say the smallest object is 3 in size in our image. If you want to go back to the non-highlighted image, you just click the back arrow here, and you can increase the size on the maximum objects here as well. And you can see that most of the objects fall below 40 in size pixels. Now you can go back to the enhance or suppress features module and change the feature size to maximum, because we now measured in the display history and measured the maximum size of the RCPs. So if you had any other maximum size of your objects in your images, then the default setting, you can change it here. Actually, the feature size default matches the example images that we have. But if you were to change the size here, I'm just going to change it now to show you what happens. Your cell profile actually moves back to that module in test mode. So you will have to click your way through the modules again to get down to where you were if you go back. So in this case, we had no change in the feature size. For any of the channels, we keep it to feature size 40. And you'll have to then click yourself through these modules again to get to the next module that we're going to look at, because we're going to look at the identify primary objects as well. So just click myself through these two to get down to the identify primary objects. The cell profile has now moved on to the identify primary objects here. Clicked myself through enhance or suppress twice by clicking step. So now we're in the identify primary object. So here, if you measured any other minimum and maximum object sizes in the display histogram, you can change it here. But the default settings matches our images, so we keep it to 3 as a minimum object size and 40. And when you press step here, the profile will show you an output window, and it will show you the segmented rolling circle products based on your thresholds. So if I again press the magnification zoom here, and I highlight an area that I want to look at here, you want to make sure that you have actually segmented or picked up all the rolling circle objects, the product objects in your image. If you are missing some, so the green means that I've actually selected those objects. If you're missing too many here, if you see objects that are not selected, then you need to adjust your thresholds again. So you might have to adjust your minimum and maximum thresholds. Another setting that could have to be adjusted is this manual threshold for the intensity. We have here we show saying that anything above 0.07 in intensity should be selected. You might, if you have very weak signals, you might have to go down to 0.05 or 0.04, for example, to select all of them. But you're aiming to select all of the objects at this stage in the image, and you can see here that these other objects have been selected, and you've selected 13,492 accepted objects in the 647 channel. And then you will do exactly the same thing for the 565 channel. Again, adjust the minimum and maximum thresholds if you have to, and you press step. There's one more thing I wanted to show. So I mentioned that if you go back to the module and change setting, I'm just going to show you now. Cell Profiler will actually jump back to that module in test mode, and that means that obviously you have to run through these following modules again to get to where you were initially before you went back. So if you don't want to just click on them individually, you could actually set a pause button on the next module that you want to look at. So here I want to look at the expand or shrink objects because I've looked at the module down to display histogram. And I don't want to sit here and click them. Then I could actually just put the pause button there and click run. And it will then run through the modules automatically down to where I put the pause button. Now we have stopped the enhance or shrink object here. So I will remove the pause button. This module, expand or shrink objects, is performed for both the ATO565 and the ATO647 channels. So we have two of those. And this module aims to shrink all the RCPs into single pixels so that they can be expanded by a defined number of pixels in the next module. So this effectively reduces the area of each RCP where fluorescence intensity will be measured. So this procedure improves RCP segmentation and therefore lowers the risk of generating false positive protein interaction results. So two closely located but separate RCPs will be less likely to be falsely classified as overlapping later on in the classify objects module. So you will use the default settings and just press step. The window looks like this, where all the original segmented RCPs have been reduced to single pixels. And these will then be expanded in the next module, which is called the Identify Secondary Objects module. And they will be expanded by a defined number of pixels here. So start by using an expansion of 4 pixels. And so an output window will be showing. I'll show you that one now. The output window will show the input RCPs, but with an overlay of magenta circles that represent the expanded area starting from single pixel object. So I'll highlight this now to show you how it looks like. So you see that the RCPs first in the previous module, they were shrunk to 1 pixel and then expanded by a defined number of pixels. And these are then the areas where the fluorescent intensity will be measured per RCP. And the aim is to choose an expansion number that generates these magenta circles that are covering as much of your RCP as possible, but without creating any touching between the circles of different RCPs. Because if you expand too much, you're running the risk of creating false positive protein interaction results if they're touching. So I'll show you 2 examples where we have expanded by 4 pixels and then expanded by 6 pixels, and there you can see the difference now. So this is 2 different examples. So this is one I showed you where we've expanded with a pixel size of 4, and you see that the circles are not touching each other too much. In this area here, this is a magnified area of the same image I just showed you. But when we expand by 6 pixels, you can see that the circles are starting to touch each other too much. So this is this means that we're running the risk of creating false positive results. So basically, you want to have something that looks a bit like this. So we'll be doing these identify secondary objects for both channels. We started off with the Atlas 647, and we will also do the Atlas 565. Choose the same pixel expansion for both channels. So in this case, we've used 4. And here are the results also for the Atlas 565 channel. So we do this for both the 647 and Atlas 565. So now we moved on to the combine object. This module combines the expanded Atlas 565 and 647 RCPs into one image to give all RCPs, or total RCPs, that we call it. Here you would just use the default settings and move on to the next module. So I click here, the green arrowhead, or step. So the next module is mask objects. So the green arrowhead is moved there. I'll just put this mask objects here to look at the settings. This module will select the RCPs that are located within the cell borders that we identified in the identify secondary objects. And it will name the total RCPs in cells. So the output from this module will be looking at the total RCPs, but within the cell borders. So we name them in cells. And here again, use the default settings and move on to the next module. Quickly before I do that, I want to mention that as we discussed earlier in module 1 and 2, if we haven't been able to identify nuclei and cell borders because of having trouble with unevenly stained nuclei, for example, then we won't be able to quantify RCPs in cells. So this could be, for example, in tissue. So I will show you how to handle that later on in this tutorial. So I'll just press step here now, because these images are cell images. So now we moved on to measure object intensity. This module will measure the intensity for the total RCPs in cells in the Atlas 565 and Atlas 647 channels. And again, here you will use the default settings and move on to the next module. The next module is called display density plot. And here, the total RCPs in cells are plotted based on their integrated intensity that was measured in the measure object intensity. So the intensities of RCPs in the Atlas 565 channel is plotted against the intensity in the Atlas 647 channel. Here again, you'll keep the default settings and click step to generate an output window displays the density plot. Click step. And here the density plot output window will display the Atlas 565 intensities on the x-axis and the Atlas 647 intensities on the y-axis. So the density plot displays the distribution for the RCPs according to their intensity in these two channels. So high density areas down here along the axes represents RCPs of similar fluorescent intensities. So having the red color here would implicate that you have many RCPs that are having these types of intensities in a relatively high intensity in Atlas 565 and a relatively low intensity or very low intensity in 647. So we use the cool warm color map here, indicated by this scale to the right. So the next module called classify objects, this is where we will use the density plot and to classify and filter RCPs based on their intensities in both channels, and they'll be classified as either Atlas 565 RCPs, Atlas 647 RCPs, or overlapping RCPs, or even background RCPs. So basically Atto 565 RCPs will be RCPs that are having intensity in the 565 channel but low intensity in the 647. And Atto 647, on the other hand, would have relatively high intensity in the 647 channel but no intensity in the 565 channel. Overlapping RCPs will have intensity in both channels and will be above these classification thresholds that we're setting. So they will be classified as overlapping. So I'll show you a couple of examples now how to interpret these plots. If we had 100% overlap in intensities, so that is, if you had RCPs that were equally bright in both the Atto 565 or 647 channels. So if you had, for example, two proteins that were all 100% interacting with each other, then we would have a linear relationship between the two channels on the density plot. It will look something like this. So the intensities of 565 and 647 will follow each other in a linear relationship. On the other hand, if we have two proteins that are localized to two different subcellular compartments, for example, and they are not interacting much at all, then we will see an exclusive relationship between the two channels on the density plot. And most RCPs will be lining the X or Y axes on the density plot. And they will be looking something like this, where you have the highest density of RCPs will be actually having only intensity in one channel and nothing in the other, vice versa. If you have proteins that are not really meant to interact. But in the example presented in this tutorial, MolBoolean was performed for E-cadherin in the 565 channel and beta-catenin in the 647 channel. And these two are interacting in MCF7 cells. But they're also expected to be present as free, non-interacting proteins. So then we'll have a density plot that looks a bit like this, where we have some RCPs that are only single or individual E-cadherin proteins, so basically only having fluorescent in the 565 channel, and also single beta-catenin proteins only having fluorescent in the 647 channel. But you will also have quite a few RCPs that are having fluorescent in both. So now we want to add intensity cutoff values in the enter cutoff value here for the Atto 565 and the Atto 647 channels. The cutoff values can be estimated based on the density plot. So if we enlarge the area down at the bottom of the plot like this, all the magnification glass, press the magnification glass, have it, and then drag down here. Then we want to draw an imaginary vertical line at a value on the x-axis where the main bulk of RCP start to appear in the Atto 565 channel. So you can see that most of the RCPs are starting to appear around here. So we draw a vertical line up. Just keep that in mind that this is where we want to be. So and the same thing for the 647 channel. You draw another line where the main bulk of RCPs are starting to appear on the 647 channel. So you remember that, so we can round up the numbers to 3 on both axes. So we put 3 as a cutoff value. So it will look something like this. This is where from the instructions for use. So this is the original density plot, and we've done the vertical lines here. So if you draw these out, these lines manually, you can see that this is where the RCPs are starting to appear on both axes. And here we would round up the value to 3 in both instances and put the cutoff value of 3 in the classify objects. So if we now look at an imaginary illustration of how the objects would be classified based on the cutoff values. They're divided into groups based on the cutoff values. So any RCPs with intensity above the cutoff value on the x-axis, but below the cutoff value on the y-axis, will be classified as having intensity in only 565 channel. So that's the E-cadherin protein being present on their own in the image. And vice versa, any RCPs with intensity above the cutoff value on the y-axis, but below the cutoff value on the 565, the x-axis, will be classified as single ATO 647 RCPs, and that would be single individual beta-catenin proteins. And any RCP that would be having intensity above the cutoff values in both channels would then have intensity in both the ATO 647 and ATO 565 channels, and they would be classified as overlapping or interacting E-cadherin and beta-catenin proteins. So if we now press step in the classify objects module, then the output window will plot the number of RCPs in the image according to the cutoff values and the classification bin names that I just mentioned. So here you will generate 565 RCPs, 647 RCPs, overlapping RCPs, but also background RCPs. And the background RCPs are the ones that fall below both cutoff values. In the illustration I just showed you, you can see background RCPs down here. So that is RCPs that are having very low intensity, so basically below both cutoff values. And it's important to remember that these cutoff values that I've just described, they are set by this manual interpretation. So it's up to the user to decide the stringency for the classification of single versus overlapping present RCPs. So, and obviously the cutoff values would ultimately affect the determined number of free versus interacting proteins in the output data. So the main important thing is that you're consistent when you're comparing maybe control versus treated samples, for example. So now we have defined all the settings for the images, and the remaining modules of the pipeline relates to the data output, and we don't need to test it in test mode. So we can exit test mode now and actually move on to the actual analysis. So you just click exit test mode down here at the left hand bottom of the page. So now we've exited test mode. So now we want to analyze the images and generate output data. Before we press analyze images down here, there are a few things that we need to do first. And the first thing is to deactivate the measure object size and shape. You just unclick that green button, click there. And also the display histogram can be unclicked. So these modules are not needed for the image analysis. They're just there to help you set the size of the RCPs. And then we want to deactivate the I symbols for all of the modules. We don't want to see the output during the analysis. It will slow down the actual analysis and could create error messages. So we don't need to see them when we analyze. We've already looked at them in test mode. So we're just unclicking all the I symbols. We don't have anything open. And then we want to save images. So we have two save images down here. The first one here, you just want to output images in a folder on your computer of choice. So click this folder icon here and place it in your in the folder of choice. But I have everything gathered here for the tutorial. You'll have something called data output, for example. You can just select that data output there, and you have another save images module. You choose the same output folder, data output there. Select folder. And then finally, in the export to spreadsheet, so this is the actual data. You want to, there's a subfolder up here. You want to select where you want the output data to be placed. So again, I put it in the data output there. And you want to name your output file. In this case, we can just name it. It's ECAD beta captaining, like that. So then we will start an image analysis by clicking Analyze Images down here, the left bottom corner of the window. And the time it takes to analyze images depends on the number of image sets that you have. To analyze 3 image sets takes roughly 5 minutes. So just to keep in mind. So I'll press Analyze Images here. And we start to analyze images. So once the analysis is completed, you go to the folder where you saved the files. So in this case would be data output, divided into cells and tissue, because I will show you tissue later as well. But in the cells, so for each image set, Cell Profiler will generate an output image file called classified RCPs. And it basically shows the distribution of RCPs in cells. I'll open it now. Classified according to the cutoff values that we set in the classify object module. So here we have the RCPs that were classified as ATO565 only, so E-cadherin, so individual E-cadherin protein in blue, light blue here in this case. And the RCPs that were classified as having intensity in only the 647 channel, so individual beta-catenin protein in yellow. And then you have RCPs that have intensity in both present channels, so overlapping RCPs, indicating E-cadherin and beta-catenin interaction in red. Then you have some dark blue objects as well that are the background RCPs. There will also be an additional image generated called original overlay TIFF. And that's basically the same as I just explained, but you have also the nuclei outlines in the image as well. You can see in green here, you have the nuclei and outlined as well. Cell Profiler will also generate 2 CSV files. I'll show you that as a list instead. here. One is named, so basically you have the naming of your file, E-catenin and beta-catenin that you indicated in the cell profile, and then it's called experiment CSV, one of them. You click on that. You basically have a list of all the modules that were used during the cell profiler run. So if you wanted to go back and check how you ran the analysis afterwards, you can check and see exactly what inputs you put in here for the different modules. The other file called E-catenin and beta-catenin image CSV, and that contains all the output data on the number of RCPs that were quantified in the different classification groups that we set. But the data in the CSV file needs to be processed slightly before you can make use of it. Because the image CSV file is comma delimited, so we need to convert the text format to columns. And I'll show you how to do that. So you open the file. And then you highlight the first column, column A, like this. Then you go to data, up at the top, and the columns. And in the window that opens, you click obviously the limited option and go next, and then you click comma here, so you have tab and comma selected, and you click finish, and then you get the column, all the information sorted into columns. If you need to, depending on obviously if you use Microsoft Office with that recognizes dots or commas, you can convert all of them to commas or dots, and if you please. So I'm just going to convert them in this case to replace all dots with the comma. Replace all. So these output columns now contain information on the number of counted RCPs in cells for each classification group. So each row represents a different image set. So I'm just going to drag these out so we can see text in each one. Also centralized. Okay, so the count after 565 RCPs are the E-cadherin proteins, so the individual E-cadherin proteins in your images. So this row here would be your image number 1, image number 2, image number 3. So I'm just talking about image number 1 now, and this column here, column B, will be your count of Atlas 647 RCPs. So that's your beta-catenin, the individual beta-catenin proteins. The count background is obviously the RCPs that are falling below the cutoff values in both channels. You have your count cells. It's the same as the count nuclei, the number of nuclei and cells identified in the image. And then you have the count overlapping RCPs. So these are the RCPs that have fluorescence in both channels based on your cutoff values. So these would be beta-catenin and E-cadherin interacting proteins, complexes. And then you have the count for total RCPs. That's all of the RCPs in your image. And then you count total RCPs in cells. So these are the RCPs that were counted within the cell borders that we selected. And then you have your image number, and the metadata in our case is the same as the image number. You could have other metadata, as I explained in the metadata module at the beginning. You now want to save this output data as an Excel workbook. You just press save, save as, and you place it in the folder for your output data. Here you can name it, so you basically save as type, Excel workbook, and you can save it as calculation summary. I already have a file here, calculation summary tutorial. So here is the calculation summary Excel workbook, and so here are the raw data at the top, and that I just showed you coming from the CSV file. And I prefer to reorganize the columns slightly in this order. So I have the image first, then I have the count of total RCPs, then count of total RCPs in cells, and then I've added an extra column that is called total RCPs in cells with background subtracted. Because we don't want to have the background as part of our calculation. So we want to subtract the background from the total RCPs in cells. So it's basically just having a count of the total RCPs in cells minus the count background. And then I have the ATO 565, 647, and the overlapping and nuclei. So I like to organize it like this. You can obviously organize it however you want to do it from the raw data. MolBoolean data can be presented as you prefer, and I will be showing two different approaches to how to present the data now. The first one is to use the information in the output table and to normalize the RCPs in the different classification groups to the count of nuclei or the count of cells in the same image. So basically, you would divide the number for each classification group with the number of nuclei. So here for total RCPs, I'm using the column where I've subtracted the background, so count the total RCPs in cells, and then divided with the count of nuclei. And I do that for each image. And then you do the same for the overlapping count. Yeah, so basically taking the overlapping count from image set 1 divided by the nuclei. So we will get an RCP count per cell for each classification group, and because we've done 3 image sets, we can calculate an average and the standard error mean and plot it like this. The second approach is to normalize the RCP count for the different classification groups with the total number of RCPs in the image. So you create fold values. And so here basically you've taken, for example, the overlapping here divided by the total number of RCPs in cells with the background subtracted for that one. And you can do that for each image set and for each classification group. And then you can convert the fold values to percentages by just multiplying them with 100. So we will then get an RCP count as a percentage of total target protein, basically, here. So we can see that 50% of the total number of RCPs in the image have been classified as overlapping. And so 31% of the total amount of RCPs in the image have been classified as having present in only the 565 channel, so E-cadherin only. Or you have 19% of them being classified as beta-catenin only. Like I mentioned earlier, cell profiler may not always be able to determine the number of nuclei and cell borders in the image sets. For example, this could be difficult in tissue images where the nuclei shape and the staining intensity are often less uniform. So here's an example of a human small intestine tissue sample where we have stained with Hoist. And you can see that the nuclei has staining on the borders, but nothing in the middle, for example, and there's a varying intensity. So I'll show you in Cell Profiler that it struggles to select the number of nuclei. So basically it gives you a number of accepted objects as 0. There may also be cases in fixed cultivated cell lines where it is difficult to determine the number of nuclei. So the following instructions that I will show you now can be used in all cases where it is difficult to determine the number of nuclei in your image. So I will now perform image analysis on such an example, and I will use MolBoolean tissue staining as an example image set. And there are a few minor adjustments that can be made in the standard pipeline that would allow analysis in tissue. And so we basically don't need to perform the nuclei and cell segmentation in these cases. So I've now uploaded decomposed images from MolBoolean tissue staining, and according to the instructions I gave you earlier. And I've started the test mode, and we now are in the Identify Primary Objects first module. So when we click Start on that module, generate the output window, you will see that the Cell Profiler struggles to segment the nuclei. I enlarge this area here. Basically, it's not selecting. These magenta circles means that it's not been selected. So number of accepted objects here is 0, and there is nothing, no nuclei being identified, as you can see up here as well in the nuclei output. So in these instances, it will not be possible to count RCPs within the cell borders. So there's no point even doing this, the identify secondary objects either, because you want to be able to quantify or segment the cells based on any nuclei, because we haven't identified any nuclei. Instead, we will have to count RCPs throughout the entire image, so not within cells. So the number of counted RCPs for each classification group can still be normalized as a percentage of total RCPs, as I explained, as one of the approaches to present the data for cells. But the only difference is that the results will represent total RCPs per image set, rather than the total RCPs in cells. We will still process the images through the identify secondary objects, but you will not use the results from this module. You just ignore the results, but we still have to click step in this one. And as you can see, no cell objects have been selected. But we don't have to worry about this, and I'll show you why. But as I mentioned, we still have to have these modules activated, but we ignore the results from them. So now you will go through all of the modules in test mode as exactly the same as explained for cells, but we just have to make a few small alterations to the module setup. So the first thing is to unclick the mask objects and to unclick the overlay outlines, and the second save images. And as you can see, this will result in error messages, and this will be indicated by these, the crosses in subsequent modules. But don't worry about this. We will now correct these error messages. The first thing is to go into the measure object intensity, and here we'll deselect the total RCPs in cells here, and instead scroll down to the end and select total RCPs instead. And these changes should remove the error message from this measure object intensity module. Next is to go into the display density plot module and change from total RCPs in cells here to total RCPs instead. So basically not using RCPs within cells, but just RCPs in the whole image. And you have to do that in two places. So both the Atlas 565 and the Atlas 647 channel. So you do total RCPs here as well for the image Atlas 647, and that should remove the error message for that module. And then you go to the classify object module and change from total RCPs in cells to total RCPs there, and the same thing. No, that's just in one place from this module. And that should remove the error message there as well. And then in the filter objects module, you change from total RCPs in cells to total RCPs. And that should also remove the error message. And then you do the same for all three of these ones. Same thing for all filter objects modules. Click on them, go change total RCPs. Total RCPs. So that should remove the error messages from them all. And the very last thing is to go to the export to the spreadsheet module. And here you need to click here, press button to select measurements. And in the window that opens, you will go to image, count, and unclick cells and nuclei option here. You don't want the output on that, but all the others remain. And then you press OK. And then the error message from here goes away, but you still have this exclamation mark, meaning that you can't produce output in test mode, is saying. So now we've done these small alterations to the modules. We can go through the different modules in test mode again, from the enhance or suppress features onwards. And basically according to the instructions I gave you earlier for cells, and you can then set the thresholds based on your tissue staining images. So I won't do that now because I know that my settings are good for tissue. So I will just exit test mode. And I will again remove the I symbols from all the modules. And I will unclick the measure object size and unclick it here with the green and display histogram. And then I will just make sure that I have the right folder. In the save images, I'm clicking here the where I want to save my images. So I'll put data output tissue, and also in the export to spreadsheet, and I will select the output folder, same place, and I will name this output file tissue, like that. And then we can analyze the images again just by pressing analyze images. Please note that sometimes this exclamation mark remains at the export to spreadsheet module, saying that it cannot produce output in test mode. Don't worry about this. You can still analyze the images. It just cell profiler sometimes lags in removing this indicator. So we can just press analyze images, and the profiler will start to analyze the images. So the image analysis takes roughly 5 minutes again, and then you will have your output data saved in the folder that you inserted. For the data processing, we can do the same as for the cell example, and there will be a slight difference in the output data compared to cells. First of all, you will not generate an image with that's called original overlay, because you don't have any nuclei selected, so you will only generate the classified image. Same as for cells. I'll open it now, and you'll see the RCPs that have been classified to different groups, so i.e. ATO565, ATO647, or overlapping RCPs, but you will not have any nuclei indicated in this image. The output data in the image CSV file can be processed as explained before, with comma delimited text converted to columns, and data output columns for tissue analysis will not include any count of total RCPs in cells. It will only have total count of RCPs per image instead, and it will not have any count of nuclei or any count of cells. The results can be presented as explained previously, but only as option 2, and that is to present the RCP count for each classification group as a percentage of total RCPs, so total target proteins per image. But the only difference is that the results are as a percentage of total RCPs per image rather than within cells. So that's how image analysis differ compared to cells, or in images where you cannot identify nuclei and cell borders, basically.